Hello again. Welcome to another Google Hangout. My name is Colin Cairns. I'm from the Baker College of Auburn Hills Learning Center, and today I am doing a Google Hangout for A&P 1, Chapter 3, Cells and their Function. And I'm going to try to get a screen share going. Apparently, it's not working correctly right now. So I'm going to try to get the screen share up again. And here we go. And okay, I believe that's the screen share that I need. So let's do an introduction to Chapter 3. The basic organizational structure of the human body is a cell. There are 75 trillion cells in the human body. The cell size is measured in micrometers. The process of differentiation is when cells specialize. As a result of differentiation, cells will vary in size and shape due to their unique function. So muscle cells will vary in size and shape from nervous system cells, cardiac cells, respiratory system cells, and so on. Now let's take a look at the composite cell in this next slide. Also called a typical cell. The major parts will be the nucleus, the cytoplasm, where all the organelles are located, and in the cell membrane. Looking from the outside in, the cell membrane controls what moves in and out of the cell. It's selectively permeable, which means it only allows certain molecules and substances in and prevents the entry of others. It's a phospholipid bilayer. It has water-soluble heads that form the surfaces on the outside, and those are called hydrophilic. The water-insoluble tails form the interior, and they're considered hydrophobic. This bilayer is permeable to lipid-soluble substances. Water-soluble substances are not allowed to penetrate the membrane. Cholesterol stabilizes this membrane. And there's also proteins in the membrane, receptors, pores, channels, and carriers, enzymes, cell adhesion molecules, and then self-markers. This next slide shows a view of the cell membrane itself. And as you can see, the heads are phospholipid heads are hydrophilic, and the tails on the inside are hydrophobic. On the extracellular side, you'll see carbohydrates, glycolipids. There's a globular protein. Cholesterol molecules are embedded in the layer. And then you can see on the inner that there's also a cytoplasmic side of the membrane. But the only thing important to know right now about cell adhesion molecules, they're important for growth of nerve cells. Now inside we have the cytoplasm. That's a network of membranes and organelles suspended in the cytosol. There's a cytoskeleton that forms a supportive framework, and this is made of protein rods and tubules. The organelles inside, beginning with the endoplasmic reticulum. It's an interconnected membrane-bound sacs and canals and vesicles. It's used as a transport system. The rough ER is studded with ribosomes, and the ribosomes are the site of protein synthesis. The smooth ER, on the other hand, has no ribosomes on it. It's a site of lipid synthesis. Those lipids will be added to proteins arriving from the rough ER. The ribosomes themselves are either free-floating or connected to the ER, and they provide structural support and enzyme activity to amino acids to form protein. Another important organelle in a cell is called the Golgi apparatus. That's a stack of flattened membranous sacs. What happens here is the ribosomes bring their proteins, and the Golgi modifies, packages, and delivers the proteins via what's known as a vesicle. The vesicles are pinched off from the actual membrane of the Golgi, and then these contents of the vesicle can be transported without any losing of the contents. And eventually, as we see in this one slide on the right, they can be secreted outside of the cell. Another important organelle is the mitochondria. And these are membranous sacs with inner partitions. Inner partitions are called cristae. The function of the mitochondria is to generate energy by breaking down a substance known as ATP, or adenosine triphosphate. This next slide shows some interaction of organelles. First, we have lipids that are being synthesized in the smoothie R. You see some milk protein genes are transcribed into mRNA, and that mRNA will exit through the nuclear pore. And then most proteins are synthesized on ribosomes associated with the membranes of the rough ER, using amino acids that are in the cytosol. Sugars are synthesized in the smoothie R and Golgi apparatus, and they could be attached to the proteins or secreted in vesicles. So proteins are secreted from vesicles that are pinched off of the Golgi apparatus or budded off. And there's just showing a milk protein in a Golgi vesicle. In the meantime, we have these lipids that are little fat globules or droplets, and they're becoming bigger and bigger, and eventually they will be excreted from the cell. Some more important or other important organelles: lysosomes. Those are enzyme-containing sacs. They digest worn-out cell parts or unwanted substances. They're important for the body's immune system. Peroxisomes, they're also enzyme-containing sacs, and they break down organic molecules and toxic substances. You'll find these organelles abundant in liver cells. Then we have the centrosome. These are two rod-like centrioles. They have two rod-like centrioles. They're used to produce cilia and flagella, and they distribute chromosomes during cell division or mitosis. And we also have cilia. Cilia are short hair-like projections on the surface of the cell. They propel substances along the cell surface. They're important in respiratory tract and in the intestinal tract. Flagellum, they are long tail-like projections unique to one cell, that will be the sperm cell, and these provide motility or movement for the sperm cell. Another organelles that we talk about are part of the cytoskeleton. These are the microfilaments and microtubules. They support the cytoplasm, and they allow for movement of organelles. They're also known as the muscles of the cell. In your book, in 3.2, there's a clinical application describes a disease at the organelle level. And let's talk about the cell nucleus now. It has a nuclear envelope, which is a porous double membrane. It separates the nucleoplasm from the cytoplasm. The nucleolus inside is a dense collection of RNA and proteins, and it's the site of ribosome production. Remember, we talked about ribosome as the site of protein synthesis. Well, in order to synthesize proteins, we first must have ribosomes, and they're produced in the nucleolus. The chromatin consists of all the cell's chromosomes, each containing DNA wound around proteins, and there's fibers of DNA and proteins in there, and they store information for the synthesis of proteins. So now we have these three main parts of the cell, and let's talk about movements in and out of this, into and out of the cell. We have two types, physical or passive processes and physiological or active processes. The passive process requires no cellular energy and includes four types: simple diffusion, facilitated diffusion, osmosis, and filtration. The active requires cellular energy and includes active transport, endocytosis, exocytosis, and transcytosis. First, we're going to take a look at simple diffusion. And all that is movement of substances from a region of high concentration down a pressure gradient to a region of lower concentration. Some of the molecules that can be diffused are oxygen, carbon dioxide, and lipid-soluble substances. So what happens is if you put a lump of sugar in water, it's going to diffuse outward from that high concentration lump of sugar and spread out into the water and try to create what is known as equilibrium, concentration the same everywhere. And this shows the solutes on the A side and solutes on the B side, and some of the heavier solutes will move to the B side, and a few of the B side solutes will move over to the A side, and eventually we will get an equal number of heavy and or bigger and smaller solutes in the solution, an equal concentration on both sides. So let's see if we can do an animation today that works actually. And no, we're not going to get one. Okay, so now we talk about this facilitated diffusion. That's diffusion across the membrane with the help of a channel or carrier molecule. Some of the things that will use facilitated diffusion are glucose and amino acids. So we have particles in a region of higher concentration. They'll naturally want to go to a region of lower concentration, but they need the help of a channel. And so they will go through this protein carrier molecule and go through the channel inside. And then they'll be diffused into the inside of the cell on the other side of the cell membrane. Osmosis, however, is the movement of water through a selectively permeable membrane from regions of higher concentration to regions of lower concentration. So what will happen is the water will move toward a higher concentration of solutes. As we see in number 1, we have a selectively permeable membrane, and we have an A and B side. The A side is more dense with solutes, higher concentration. So water will leave the B side and cross the membrane into the A side. And the water will increase on the A side to match the concentration of solutes. It'll decrease on the B side to match the same concentration of solutes. So A and B will have the same concentration. Now let's talk about osmotic pressure. That is the ability of osmosis to generate enough pressure to move a volume of water. And osmotic pressure increases as the concentration of non-permeable solutes increases. So if you have a real higher concentration on one side of a barrier or membrane as opposed to a lower concentration on the other side, you'll have some pretty big osmotic pressure differences, and that will allow the movement. So these 3 slides, A, B, and C, illustrate a red blood cell, and what you have in the first slide is isotonic. So the cell is placed in isotonic, or the same pressure as the cell themselves, have no net gain or loss of water. They will remain the same. If you place a red blood cell in a hypertonic solution where there's lots of solutes, that cell, the water from the cell, the red blood cell, will leave and move to the higher concentration of solutes. So the cell will lose water. If, on the other hand, you place the same cell in a hypotonic solution or less solutes than the inside of the cell will take on water. It will gain water. So it's important to understand, if you put a cell in hypertonic, it loses water. If you put a cell in hypotonic solutions, they gain water. And then we have filtration. That's the idea of smaller molecules forced through porous membranes. Larger molecules are left behind. Sort of like filtering sand. If you put sand in a filter in chunks of dirt, the sand will go through, the chunks of dirt will be left behind. The kidneys operate in the same process. Hydrostatic pressure is important in the body. The pressure on the capillary walls keeps them intact, and the molecules leaving the capillaries will be the small molecules. The larger molecules will stay behind. And now we're going to talk about active transport. Carrier molecules transport substances across the membrane from regions of lower concentration to regions of higher concentration. Substances normally go down the pressure gradient from high to low, but in this case, there are certain times when substances need to be transported from low to high, so that requires energy. To go against the grain requires energy. And some of the substances that will go through this process are sugars, amino acids, sodium ions, potassium ions, and other ions. And we can see by the slide, we're using a transported particle. It's in a region of lower concentration. It's going to go through the phospholipid bilayer, but it does need a carrier protein. It needs to bind in a carrier protein, but to be pushed through the channel, it's going to require cellular energy to push it through. I don't believe you have gone over the sodium-potassium pump, so we'll skip over that. And this is not going to work, so that doesn't matter. Let's talk about endocytosis. The process whereby cell engulfs a substance by forming a vesicle around the substance. The substance attaches to the cell membrane, and the membrane pinches off inside the cell in the form of a vesicle and can transport the substance around inside the cell. We have three types: pinocytosis, when the substance is mostly water; phagocytosis, when the substance is a solid; and receptor-mediated endocytosis. That requires the substance to bind to a membrane-bound receptor. And this first part shows the phagocytosis process. The particle attaches to the membrane, and a vesicle pinches off after it surrounds the membrane, pinches off this particle into the cell. And then you have receptor facilitated, where the molecules outside the cell attach themselves to receptor protein, and they usually have to have the same, like a lock and key. It has to fit, and then the cell membrane indents, and a vesicle forms and pinches off into the cell. And then there's an exocytosis, which is just the reverse of endocytosis. Cell digestion and then releasing waste products. Substances in the vesicle fuse with the cell membrane, and then the contents are released outside the cell. And another way cells use exocytosis is when nerve cells are released from, or neurotransmitters are released from nerve cells and cross over to stimulate muscle cells. And then transcytosis is this endocytosis followed by exocytosis, transporting a substance rapidly through a cell. And an example of that would be HIV crossing a cell barrier, cell layer. The HIV part of it, we have infected white blood cells. There's viruses. The blood breaks off. It undergoes receptor-mediated endocytosis. A vesicle surrounds it, and then it goes right through the lining epithelial cells, and it's released on the inside, and then it goes to infect other white blood cells on the other side of the lining. Now we'll talk about the cell cycle. So these are the series of changes the cell undergoes from the time it forms and the time it divides. We have interphase. We have the G phase of cell growth, the S phase of genetic material replicating, and a G2 phase. And then we actually start with mitosis, and we have the prophase, metaphase, anaphase, and telophase. And then during mitosis, we start the process of cytokinesis, or the division of the cytoplasm. And at that point, the cell will grow, and there's a restriction checkpoint from there on. Either the cell is going to remain specialized, or the cell is going to undergo programmed cell death, which is called apoptosis. Okay, interphase, it's very active. The cell grows, the cell maintains routine functions, the cell replicates genetic material to prepare for nuclear division. The cell will synthesize new organelles. Like I said, the G phase, the cell grows and synthesizes structures other than the DNA. And during the S phase, the cell replicates DNA. Now we have mitotic cell division. Two daughter cells are produced from an original somatic cell. The nucleus will divide during mitosis, and the cytoplasm divides during cytokinesis. The phases are prophase, when the chromosomes form and the nuclear envelope disappears. The metaphase, where the chromosomes align midway between the centrioles. The anaphase, when the chromosomes separate and move to the poles. And telophase, when the chromatin forms and the nuclear envelope forms. The mnemonic that's used to remember these phases in order is passed my anatomy test. Prophase, metaphase, anaphase, telophase. And this slide shows we have the nuclear envelope, the chromatin fibers, and the centrioles. And during the late interphase, the cell has passed restriction checkpoint and completed DNA replication. And now, as it enters prophase, the chromosomes condense and become visible. The nuclear envelope and nucleolus disperse or break down, and the spindle apparatus is formed. The centrioles separate. And during late prophase, the centromeres start to align a little bit. The spindle fibers stretch, and the centrioles head each one to different poles. Then, during metaphase, those chromosomes align along the equator. And in anaphase, the sister chromatids separate to opposite sides of the pole. And then we start the events that begin leading up to cytokinesis. And during telophase, the nuclear envelopes begin to reassemble around two daughter nuclei. The chromosomes decondense, the spindles disappear, And we had the division of the cytoplasm into two cells. And then a cleavage furrow is formed as the nuclear envelope continues to form. And eventually that furrow breaks apart, and we now have two daughter cells. And this is going to be a time of normal growth and function, the interphase. And again, this will butt up against a restriction checkpoint. Either they'll continue to become specialized, or they'll go through programmed cell death. So the cytoplasmic division is known as cytokinesis, begins during anaphase, continues through telophase, and then eventually those contractile rings will pinch the cytoplasm in half. And again, I can't get these to work, unfortunately. I don't think we did too much about control of cell division. Tumors are the obvious consequence of a loss of local cell cycle control. There are two types of tumors, benign and malignant. And that is one of the functions of apoptosis, is to regulate the number of cells and prevent tumors from forming. And I don't think we covered this. So there's apoptosis again. A little bit of what happens. Enzymes called caspases destroy various proteins and other cell components. Cell becomes deformed, turns into blebs and cell fragments. And phagocytic cells engulf cell remnants, and the cell components are degraded. Eventually, as shown here, this is the epidermis peeling away from someone who has had a sunburn. Okay, so these are the important points that you need to know. Differentiation, how cells differ from one another, having specialized function. Should be able to describe the three parts of a composite cell: the nucleus, cytoplasm, and the membrane. Describe each of the organelles we mentioned, the lysosomes, the ribosomes, peroxisomes, centrosomes. Describe the cell nucleus and its parts and the function of the cell nucleus. Explain how substances move into and out of cells, the difference between active and passive transport, the types of transport, from simple diffusion all the way to active transport. You have to be able to describe the cell cycle, beginning with interphase, and then the mitotic phase, and then the cytokinesis. And at least understand about apoptosis and how it controls unwanted cell division. And that will do it for chapter 3. The reason I did this one today is because yesterday, I could not get the PowerPoint to work properly. So if you watched, you were just looking at me talking. So this is a companion to the second half of yesterday's Hangout, where 4B was discussed. Okay, good luck with your tests. Have a great weekend, study hard, and I'll see you in 2 weeks. Thank you. Bye.